Hello, my name is Kim Cardoza from RTG16 from Raleigh, North Carolina, and welcome to our special newscast that will be revealing to you the ins and outs of this zombie apocalypse that has just occurred not even 1 week ago, outbreaking in Sacramento, California last Monday. We fear that this infection is going to be spread throughout the United States very, very rapidly if we do not act upon it now. Studies show that this infectious disease may even cover the United States within 1 month. We've been lucky to receive special information from our protein specialist and epidemiologist. So now we will go on to see Alexis Ledlow explain to you how protein synthesis actually works and how this is contributing to the zombie apocalypse.
Hi, I'm Alexis Ledlow, your protein specialist from the University of Los Angeles of California, and I'm here to explain to you how normal proteins are made and what can cause these to malfunction. So there are two very important processes when it comes to understanding how proteins are actually made. They're known as transcription and translation. Transcription occurs in the nucleus, which is where the DNA is located, and the DNA is extremely vital to understand how this protein synthesis is actually going to occur. So transcription is the process that enables an mRNA, which is also known as a messenger RNA, to make a copy of the DNA strip. In order to do so, an enzyme such as RNA polymers will attach to the gene and cause it to unzip, then causing a complementary base pair to take place, which creates a transcript, hence why it's called transcription. After it unzips, the mRNA, which is again the messenger RNA, will make a copy in groups of 3, also known as a codon. The double helix structure of the DNA will then zip back up, and then we get into translation. So translation is the next process, which is where the copy of the DNA will travel through the nuclear pore, which is around the nucleus, and into the cytoplasm of the cell. The tRNA, also known as transfer RNA, will locate an amino acid and connect with the copied gene plus the codon inside of the ribosome, which is where the protein is actually going to be made. So after they're paired together, the protein is made and the amino acid will be dropped off into the rest of the cell, and the protein will then go to the Golgi body where it will be folded. So just so we understand, the key organelle in this actual process is the ribosome, the nucleus, the Golgi body, and of course the DNA. So this is where we get into prions. And what a prion is, it's actually a misfolded protein which causes a malfunction, which is why these zombies are actually being made. Okay, so there are many theories as to how this prion is being made, and one of them is just that the Golgi body is misfolding the protein. But with something like that, if a Golgi body isn't going to be understanding or hasn't seen this type of protein before, it's not going to know how to fold it, so we can't really rely on that theory as much. The other theories are known as insertion or deletion. And so insertion is just described as when it's adding nucleic acid to the strand or the codon, because occasionally the RNA polymers will misinterpret the DNA, and any insertion inside the DNA strand will cause it to move down, and there's always going to be one left over, and it'll cause the DNA just to malfunction. And a deletion is pretty much what it means is that it's just going to be deleting one of the strands inside of one of the acids inside of the DNA. But with something like this, as I said before, with insertion or deletion, it will cause the Golgi body to not fold it whatsoever. So some reason that these prions are being made is probably going to be part of the Golgi body or something of that sort. So I really hope that this helped you understand how proteins are made and how this is vital into how these zombies are occurring. So thanks, bye!
Thank you, Alexis Ledlow, for that enlightening information. Here we go on to Kelsey Escourse, who is an epidemiologist at Brown University, to explain to us how this zombie apocalypse was probably caused. Hopefully this will put you at ease and give you some background knowledge of what we're actually dealing with.
Hello, my name is Kelsey Escourse and I'm coming from Brown University. I am an epidemiologist, meaning that I specialize in investigating infectious diseases. I'm here to explain various ways that proteins could be affecting the Golgi bodies within our cells through infections caused by bacteria, viruses, fungi, and protists. Diseases can occur inside an organism by pathogens, which are agents that cause disease. Pathogens can be infectious, meaning that they can spread diseases from person to person, which is known as transmission, or non-infectious, meaning that they cannot spread diseases from person to person. Pathogens include viruses, bacteria, fungi, and protists. Scientists are currently hypothesizing that viruses are the cause of this new zombie disease. Viruses, which are tiny infective agents, are probably the most plausible infective agent in this research, and they affect the cell in two ways: the lysogenic cycle and the lytic cycle. In the lysogenic cycle, viruses attach to a host cell and insert their DNA and sometimes their RNA into the host cells, causing an insertion mutation within the host cell's DNA. This viral DNA or RNA will lie dormant while the cell continues to reproduce, which copies the viral DNA into the daughter cells. This is when the lytic cycle will start. The viral DNA or RNA will awaken the cell and take over the enzymes, which will make copies of the viral DNA or the RNA. At this point, the new viruses can either violently break out of the cell structure or pinch out of the cell membrane, and they will go and infect new cells. Bacterial, fungal, and protist diseases cannot be the cause of this disease because this disease involves the Golgi body, but these pathogens can never enter the cell, meaning that it cannot touch the Golgi body inside of the cell. The virus is the most plausible of the infective agents because they can directly affect the cell's DNA, and no other pathogens can do so. Since the Golgi body is a packaging system and the way that proteins are packaged in the Golgi body depends on the protein's instructions, it is very likely that the Golgi body is misfolding the proteins because of the wacky viral instructions of the protein, which is made just like viral DNA. These misfolded proteins become prions, which causes zombie infection. Therefore, this disease could be very much like rabies, which prompts violence from the infected. In order to cure this disease, civilians would receive antivirals or vaccines. Antivirals will completely kill the virus in a way that is similar to how antibiotics kill bacteria. Vaccines include tiny, dead virus particles that are inserted into the bloodstream. This immune system will attempt to fight this virus, creating white blood cells that will be prepared to fight the real disease should civilians ever come in contact with it. That's all I have to say. So stay safe from zombies. This was Kelsey Escourse coming from Brown University.
So fellow viewers, I hope this put you at ease and this information was very, very helpful. We'd like to tell you to shut your doors and make sure you get good night rest because we don't know what's going to be happening tomorrow. We are also going to be updating you periodically of this zombie apocalyptic mess, and hopefully we'll be able to solve this mystery soon. Signing off, Kim Cardoza.